charge will be absorbed to your sample, and the charged sample will move through this top tube, and you will get the spectra. Here you can have plus points, minus points compared to HRMS and all. Here at a time within 1 hour, you can analyze 364 samples. Same sample, if you want to analyze any HRMS as a time gradient time, if you keep 20 minutes, 30 minutes gradient time, 364 sample, it may take weeks time. But same thing as a automation in MALDI TOF technology, 360 samples, you can analyze less than 1 hour. And you need only 1 microliter of sample. That's why in all central facilities, if they are having QTOF, triple quad, that is different, but every central facility, they will have one MALDI TOF technology. Because if you use any HRMS technologies like LCMS, QTOF technology, Arbitrum technology, you may have some limitations, like you can't do polymer analysis. Why? Because it will carry different contaminations. It may contaminate your tubings, everything. But here in MALDI TOF technology, you can do polymer analysis also. You can do multiple applications in MALDI TOF technology compared to HRMS. And your consumption of solvents will be less time. It will take very less time compared to our regular HRMS and all. So these are the different instrumentation spot. If any slide, if you get any queries, please just stop me and we can discuss. This is just for one example I want to explain QTOF. See, as I mentioned here, this is the inlet. Here I taken LC as a front end. This is the inlet, ESI. Glass capillary tube is there, where your ions will move through this glass capillary tube. Funnels will be there, where they will make your ions. When you are spraying, automatically as a sprinkle, your ions will move here and there. When you are having these funnels, you may cover complete, where it will give a chance for sensitivity. Just let me show this one. See, when your ions are moving like this, here, when you are spraying through ESI, through glass capillary, your ions will move. All 100% your ions will be collected from this glass capillary. So from here, first quadrupole, where you will filter your scan range as per your mass range requirement of your sample. Second quadrupole, where you will fragment your molecules. Here you are having TOF tube. Based upon your mass range, exactly it will hit the detector and you will get the spectra. This complete setup will be works under vacuum. All HRMS will works under vacuum 10 to the power of minus 6, 6, 7. Sometimes it may reach until 10 to the power of minus 9 also. This is a clear cut. What are the main factors in mass spec? We can make it as a 3 steps. One is sample preparation, another one is equation part, next one is data analysis. The main point, 80% of our mass spec analysis will be depends upon sample preparation. How much pure your sample? Is there any other solvents or buffers which are affecting your ionization part? Normally in institutes and all, the main challenge we face sample preparation part, where just we are bothered about maximum about spectra, but first we have to work more on sample preparation. Why? Because currently whatever vendors whoever is supplying mass specs and all, they are coming with dedicated ready-made methods. So here we don't need to work more on methods. Even for data analysis also, we are having different ready-made tools. Previously you used to take raw spectra, used to sit on a number of hours to just identify each peak step by step. It used to take more time. Now we are having different MS tools where directly you can upload your data. Directly it will give the result as a protein identification with the complete spectra, sequence, labeled sequence, everything it will give. Before that one, we should be very careful when you are preparing sample preparation. Next one is equation part. Once you are having very good sample, yes, I prepared nice sample. I done the digestion part, protein. I got very good peptide, number of peptides here. Just I want to confirm my protein sequence now. I have a very good sample. I filtered properly. Everything is done. You should know exactly what is the method you have to use for the particular sample. If you are using any high mass, for example, if you are using any intact level mass, protein mass about 150 kilo and all, you should know what is the scan range you have to keep for your particular mass spec model. Or just please note it. Every mass spec will have particular limitations, different parameters. For same intact mass 150 kDa, 150,000 Dalton, when you are analyzing in Arbitrap, when you are analyzing in QTOP, we can't expect same temperature, same pressure, same. It will be completely depends upon different model, different vendor. So you should know exactly what kind of parameters you have to keep for intact mass level, subunit mass level, or PMF mass level. Nothing but 150 kDa range or 50 kDa range or 2000, 5000 mass range. Okay, good. You acquired nice data, around 1 GB, 2 GB data you acquired. If you are not presenting your data, if you are not confirming your data, if you are not concluding your data properly, then there is no use of your raw data. You should have proper MS tools to confirm your protein sequence, to confirm your data. For this one, you should know exactly, these are called as processing method, data processing method. You should know exactly what are the processing methods you have to use for PMF level, peptide mass fingerprinting. If it is a peptide level, what are the parameters you have to give, threshold you have to give, what are the peaks you have to select, at what range signal to noise ratio you have to keep. If it is a protein level, you will have only single compound. If it is a protein level in your sample, you will have maximum 1 or 2 proteins. In that case, filtering is very easy for you when you are doing data analysis. At the same time, if you digest that particular protein into peptides, at least you will have 150, 200 peptides. You should not miss any low intense peptide peaks. At the same time, you should not encourage any noise peaks. So when you are processing your data, you should know the exact threshold limitations. Same thing will applicable for equation also. Simply you can't do all peaks for MSMS fragmentation. You are just going to sit on some scrap data. You should know exactly what is the concentration of your sample. Basing upon concentration, you should give, this is the noise level mass range you want to filter it. This is the mass range exactly threshold you want to go, you want to make it for fragmentation level. Simply if you acquire complete data, MS level, MSMS fragmentation, it may generate 4 GB, 5 GB data where you will waste your time more than 1 week, 2 weeks simply to analyze the data. These are the key terms you should understand when you are analyzing any of your samples. First thing, resolution. As I mentioned in previous slides, how much close species also your mass spec should able to separate it. That much good you will get the quality data. If it is a normal regular mass spec and all, just for your MS normal intact mass confirmation, that is fine. But when you are analyzing any closed species, for example, if you done the digestion, you got the very closed peptides. One is around 200.25, another one is around 200.45. That much closed species also, your mass spec should able to separate it. You can say like, yes, I'm already having front end LC. In LC, it will get separate. Yes, fine. That is fine. It may get separate in LC itself as a RT, retention time. But when you are checking any PTM studies, for example, you are having one peptide, 2 different sites. Maybe you are having oxidation in another site you are expecting, but this oxidation part is happening in different site. Your mass not going to change. Why? Because complete peptide oxidation is happened, mass will be same, but you want to check site specific oxidation exactly where it's happened. In that case, your instrument should able to separate, even though if it is having same mass, due to, I mean, difference upon site specific, your instrument should able to separate it. Maybe you can say like, still it will have same mass, same masses. No, currently we are having those technologies. Basic upon site specific also, your ions will get separate. Next one is mass accuracy. How much close your theoretical mass to your experimental mass, that one is depends upon your instrument mass accuracy. You should do continuously, I mean regularly calibration. You should maintain your mass spec properly, then only you can confirm your mass accuracy. When you are not doing your regular calibrations and all, automatically you will get some mass errors. You don't know this is happened because of any modification in your sample, or this is because of your instrument error. For that one, regularly you have to do calibration. You have to cross check your mass spec with your internal standard or external standards you will have commercially. Third one is mass range. Every mass spec will have particular mass range. For example, if you take Malditop, In multi-top, you can go until 700 kDa, 800 kDa. If you take triple quartz, you can measure nice data and all until 2000, 3000 Dalton only. Maybe maximum you can go to 5 kDa insulin protein, but you can't cross more than that. But if you take timsTOF or QTOF technologies, you can go until 200 kDa. Here kDa but 1000, 200,000 Dalton you can go ahead. So depends upon your application, you have to select the instrument where it will cover all these criteria. Sensitivity. Every time you won't get high concentrated sample. Sometimes when you want to check any impurities, for example, in biopharma companies, always we are bothered about that 99% protein. But when you go for any filing, documentation, FDA, first they will ask impurity documentation, where you have to check your percentage of that impurity level 0.002%. So your instrument should able to identify your major molecule, which is 99.9%. At the same time, it should not miss any impurities which are present 0.002%, 3% like that also. That much sensitivity your instrument should have. Here, I want to add one point for mass spec users. We should not load always high concentration samples to your instrument. Your detector will get saturated. In future, if you want to do any lower concentration sample, in that case, you may face the challenge. When your vendor is saying your instrument is sensitive for 0.5 mg per ml sample or 0.2 microgram, 0.3 microgram, please try to load only that level concentration samples only. Simply to get high concentration, high intense peak and all, if you start to using 0.5 mg, 1 mg and all, in future, when you are having lower concentration samples, you may face the challenges. Okay. Third one is scan speed. Your instrument should have the capability to check, to scan all kind of molecules. For example, at a time, in nanosecond time, if I'm releasing one molecule, your detector will identify very nicely and you will get very nice peak. But here, when you are analyzing any proteomic samples or when your gradient, that LC gradient is very, very less, automatically within less time, 0.1 minute, 0.2 minutes, 0.01 minute, at a time, you will release number of 5 peptides or 6 peptides. Your instrument detector should able to identify all 6 peptides, even though if you are releasing within nanoseconds. Here scan speed also, as I mentioned in the previous slide, equation, you should know exactly. If you are analyzing only intact level mass, that's fine. You can reduce your scan speed. Why? Because in your sample you are having maximum 2 or 3 proteins. But sorry, can I continue?
Yes, sir. Please continue. I think
Yeah.
someone has
Sorry.
unmuted the mic.
Yeah, fine, man. That's it. Yeah. For example, if you are analyzing any peptide masses and any proteomic samples, within 0.1 RT time, it may release 2-3 peptides at a time. Those 2-3 peptides will go for fragmentation at a time. Your instrument should able to identify all peaks. Okay, that is scan speed. This is a simple example for resolution. How much good your mass spec, that much nice spectra, that much nice photo you will get it here. These are example for our spectra mass spec. If your resolution is very, very less, you can't separate the peaks. These are the different stages of your resolution. Very low resolution, some average resolution, very good resolution, where you can separate every closed species into as a separate peak. This is a regular example I used to give for students when they ask about what is the overall for this all factors: speed, resolution, sensitivity, accuracy. Here you can see as a traffic signal, you are the person who is standing here as a detector. At a time, you have to scan all these vehicles. You may face big vehicles, large molecules. At the same time, you can see both cars are looking like same. As I mentioned, very closed species. At the same time, you should not miss any impurities, nothing but your small, these two-wheelers. Once, if I release this traffic signal as a green color, at a time, all these vehicles will move at a time. But still, your instrument as a detector, you are standing here and you have to check every vehicle you have to check. I may ask you once those all compounds are moved from this detector, simply I will come and I will ask you how many cars you counted, how many vehicles you counted, what kind of vehicles those are, what are the specifications. You should able to identify every molecule, every vehicle you should be able to identify. You should not say like some vehicles are moved very fast. I am busy with checking this yellow color box. No, you should able to identify bus, car, vehicle, small two-wheelers, cycle, everything. Your instrument should have that much speed. Even though if you release at a time all kind of different mass ranges samples also, I mean species also, still you should able to identify all. That is nothing but your scan speed. You should able to identify every vehicle, what is the color, what is the model, what is the company. That is your resolution. And third one is sensitivity. You should not be busy with identifying only cars, and you should not leave the two wheelers. And mass accuracy, you should not miscommunicate to me one car instead of other car. This much accuracy your mass spec instrument should have. Nothing but your HRMS, high resolution mass spec. You should not, even though if I increase the speed, you should not miss any vehicles. Even though if I say what is the exact specification, what is the color of that particular yellow color vehicle, what is the model, everything you should be able to say. Those are nothing but your peak specifications. Exactly. Every molecule will release the fragment. Still, you should be able to identify every fragment. Okay. You should have speed, resolving power, and accuracy. What is the ideal mass spec should have? Normally in commercial way, we will say like it should be low, it should have very fast identification. At the same time, it should have little lower cost. And you should get very good quality data. So till now, we discussed about instrumentation part of mass spec. What are the key factors, how mass spec will work. Now we will discuss about pharma, biopharma, the role of mass spec in pharma, biopharma. Do people have any queries, or can I continue? Regarding mass spec instrumentation part, do you have any queries?
You can continue, sir.
Yeah, thank you. So till now we discussed about different models of mass spec, what kind of mass spec for which kind of sample analysis we have to use. Fine. What is the role of mass spec in our pharma, biopharma? Hope in this last 2 years, complete world could have understood what is the importance of our life science, the importance of vaccines, antibodies, all these mostly this paracetamol. Everybody will remember now paracetamol, what is the importance. Okay, so here you can see different how can I make this one? Yeah, see here you can see different ranges of masses where you will analyze in your life science in mass spec. Aspirin, some normal, if you take any paracetamol, any chemicals, I mean we will call it as a pharma products. This regular tablets, whatever you are taking, aspirin or paracetamol, those are all lower molecules. If you come little more extent, you can see here insulin, very regular biopharma product where complete world is towards running towards insulin marketing here. That is 5,800 Dalton. If you come little more high, you can see here myoglobin, BSA, you will have, sorry, myoglobin, any other molecules where you will have 20 kDa, 18 kDa molecular weight. If you come little more forward, you can see here MAPS, nothing but your IgG molecules, antibodies, where the mass is around 150,000 Dalton. As a general code, we will say it as a, when your mass range is increasing too much, the mass accuracy will come down. Every vendor will have some mass accuracy level for every model. They could have tried it at particular mass, at particular sample. Exactly. They can't say exactly 150,000, what is the mass accuracy and all. So when your masses is increasing step by step, your mass accuracy error will be more. Why I am mentioning this error will be more, we will discuss in next slides. I'm continuously saying like MS, MSMS, fragmentation, everything. For example, if you take your protein intake level, you will confirm your protein mass, that's fine. But when you want to confirm protein sequence, you have to digest the protein into peptides. Even though if you digest into peptides, those peptides also as MS level, you will confirm it. But still each peptide I will take it, I will make it into fragments, MS level. So I am making your protein into amino acids. From amino acids, again, I will build peptides. From by using these peptides, I will build the protein. So I am not conforming intact level like 150 kDa, 200 kDa. I am not conforming. I am breaking this 150 kDa protein into 200, 300 peptides. Again, I am rebuilding as a J2A approach. Hope you won't get much confused in this coming 2-3 slides. When I'm saying peptide, your protein will break into peptides. This all just please concentrate this coming, this 2-3 slides, please concentrate here. You can see here, this is one example of one peptide. You can see R1, R2, R3, R4. I am having 4 amino acids, which is maintained as one peptide. When I am doing fragmentation of this peptide, if I am applying CID energy, collision induced dissociation energy in the presence of nitrogen, it will generate B ions, Y ions. Nothing but it will break the ions exactly near C bond and N bond, nitrogen bond here. For example, if I apply methane gas, that is called as ETD, electro distribution energy here, where it will generate Z ions. You can see here Z ions. Automatically bonding will break between this functional group and nitrogen group here. Why we need CID? Why we need ETD? For your regular protein sequence coverage and all, CID is more than enough. That's fine. You can make it. But when we need ETD technology, when we need ETD, ETD is required. You can observe clearly here, exactly it's breaking near functional group R2. When you want to analyze these proteins, whatever we are discussing here, they will have glycan bonds. Different glycan bonds will have. When you want to analyze about glycan bonds, you have to use ETD. CID fragmentation is little rough fragmentation compared to ETD fragmentation. When you are applying CID fragmentation, automatically any glycan bonds are there, it will break it. You will get only amino acid fragments. But when you apply ETD fragmentation, these glycan fragments are not going to break. It will break only amino acid bonds. It's not going to break your glycan bonds. So in that case, when you want to analyze site-specific glycans, you need ETD, methane gas. Okay, that is the difference between CID, ETD. CID just for our regular fragmentation and all, you can check it, protein sequence coverage. But when you want to analyze site-specific glycan bonds, different glycans, you need ETD technology. At the same time, in pharma companies, biopharma companies, as a safety departments and all, they won't prefer methane gas and all. That is why, because it's a safety issues. Here, this is a clear example. I want to mention it here. What is this B ions, Y ions? Why you have to confirm? How you are confirming by B ions, Y ions? What is the fragmentation here? For example, I am taking A, B, C, D as one peptide. At the same time, below I am taking E, F, G, H as another peptide. Both peptides are having same molecular weight, 100 Dalton, 100 Dalton. But the amino acid sequence is different. Here 26, 51, 23. But here 21, 4, 45, 30. Completely inside amino acids are different. But if I want to confirm MS level, I can't differentiate between ABCD peptide or EFGH peptide. So I have to go for fragmentation. When I do fragmentation, this ABCD will break in. This is, we will call it as an N terminal, C terminal. Okay, it will start breaking from, if it is a Y ions, it will start from C terminal. If it is a B ions, it will start from N terminal. You can see here, when from N terminal side, normally in protein sequence, I hope you know N terminal, C terminal sides, same thing will continue for all peptides here. So for example, this ABCD peptide, I am doing fragmentation. What it will generate? If it is a B ions, it will generate A, AB, ABC, ABCD. A and ABCD is the default. Why? Because ABCD is 100 Dalton. At the same time, from C terminal, we can say it as a to Z, Z to A approach. N terminal, C terminal. Okay. For example, if you take Y ions here, from C terminal it will start D, DC, DCB, DCBA. Okay. Same thing, same fragments. If it is coming here, you can see, you can observe here, completely mass list will be different. 25, 70, 75, 79 as a fragments. Same mass as a MS 100 Dalton, but if you do fragmentation, you can observe here 26, 74, 77, 80. So by doing this fragmentation, you can confirm this peptide is ABCD peptide or EFGH peptide. At the same time, you can confirm two sides. As a to Z approach, you can confirm, Z to A approach, you can confirm. So here you will get B1, B2, B3, 4 B ions. The same time, Y1, Y2, Y3, four Y ions. Total 8 fragments you have to get theoretically. But as a fragmentation process in instrument, maximum if you get 4 or 5 fragments also, that's more than enough to confirm your peptide. You can observe here, this is one clear peptide, how it will create Y ions, how it will create B ions. Y1 means only K. You can see from C to N it's going, Z to A approach. Y1 means only K, Y2 means KG, Y3 means KGW. Like that, as a Z to A approach, from C terminal to N terminal, each fragment will form here from Y1 to Y16 level. Around 16 fragments you will observe in Y ions. Same thing will come for B ions. You can see as a G, GL, GLS, GLSD, GLSGE. You can see here B1, B2, B3, B4. Like you will get B ions, Y ions in a single peptide. So you are confirming from N terminal to C terminal. At the same time, you are confirming from C terminal to N terminal. Like this, each peptide you will confirm fragmentation level. Then you will connect each peptide. So you will confirm your protein sequence. So intact level is direct protein sequence. Peptide level means only MS level peptides. Amino acid frequency level, sequence level means MS fragmentation level. As a table form, you can see here sequence from GL to B ions and Y ions. As a spectra, you can see here GLSD is a B4, GLSDG is a B5. Like it will continue until 16. B16 will continue. Same spectra in reverse, you can observe Y ions also. Y3, WGK, we have to call it as a KGW, KGWV, like this. In C terminal to N terminal also, you will confirm it. When you are analyzing any of your samples in mass spec, you should remember these all 4 steps. Your sample analysis starting to is not acceptable. This good precision, pure accuracy, or good precision, actually good precision with good accuracy is the ultimate we need. Okay, starting if you are doing without calibration and all, if you are analyzing your sample, you may get poor precision and pure accuracy. Accuracy is nothing but what is the comparison, what is the close from theoretical mass to your experimental mass. Precision is nothing but reproducibility. How many times you are doing, you should get same result. Here you are not getting precision also same, or accuracy also not same, you are not getting. Here accuracy is good, exactly, it's coming in, it's reaching in middle, but your precision is bad. Why? Because you can see whenever you are repeating same experiment, it's giving different precision. In third stage, you can see good precision, means reproducibility is there. For this one, clear example is our clock. Even though if your clock is on repair, every day at least 2 times you can see correct time, right? We know your clock is not working, but still 2 times you can see correct time. Why? Because it's got stuck somewhere. Like this, here your mass accuracy is not correct, that already we know, but your precision is good. Multiple times you are analyzing same sample, but it's reaching a same point. Means, for example, your wall clock is giving 5 minutes late. That's fine. You already know your wall clock is giving 5 minutes late every time. So whenever you are checking your time, you will count yourself internally as 5 minutes. Same thing here. Your accuracy is not good in your instrument, but your precision is good. Means how many times you are acquiring same data, it's giving same result. Even though it's some accuracy, mass accuracy error is there, but still it's giving reproducibility same. The fourth one, which is the ultimate one, what we expect one ideal mass spec. Your precision, how many times you analyze same sample today, tomorrow, day after tomorrow, it should give same result. And accuracy, it should match with same theoretical mass. Normally in pharma companies, where our mass will go and sit, in which lab it will go and sit. It will start from raw material. The main plus point for mass spec technology is now it's not limited for any pharma or biopharma or any environmental studies. Wherever science is there, there are mass spec technologies are there now. If you discuss in pharma companies from raw material to start, intermediate, API, active, intermediate, every step, whichever step you are doing any reaction, you have to take the sample, you have to analyze in mass spec. These all departments need mass spec where they are when they are confirming any molecule mass. If you come to biopharma, already I hope you already know the central dogma from where from DNA to RNA, RNA to protein, where you will produce your proteins. So when I am saying clone selection, here we will have different cultivations, bacterial cultivations, we will have tissue cultivations also we will have. So where our mass spec will sit in biopharma. From clone selection to scale up, final product, QC, QA documentation, everywhere our mass spec is required to confirm that particular protein sample. For example, if you take in clone selection scale up, to identify the mass, to check any modifications are happening in that particular mass and all we required, and glycan profiling, we have to check subunit level. For that one we need mass spec. If you go to final product, once you confirmed in clone selection, once you check it in 5 liters, 10 liters, then you will go to bulk production where it will be 1000 liters, 2000 liters. In that case also, still in every step you need mass spec. Why? Because when you are working in clone selection or when you are working in 1 liter, 2 liter scale up, it may not go for any modifications, PTM, post translation modification. But once if you introduce to any bulk fermentations, high cylinders like 1000 liters, 2000 liters, there is a chance of any, because of foam formation, any other quality issues may happen. So in every step you need mass spec to confirm your sample. Here, as I mentioned in previous slides here, when you are confirming your protein masses, you will have 3 steps. Intact level confirmation, nothing but we will call it as a top-down sequencing. Bottom-up mass confirmation, where we will break complete protein into peptides. Otherwise, we will have middle-up confirmation, where you will break your intact protein into subunit level. For example, if you take here antibody, you can see here light chains, heavy chain. This is middle down level. If it is an intact map, it's of 150,000 Dalton subunit level, or completely protein digestion level, where you will get directly peptides. Why can't we confirm our protein in intact level? Why we are going always for protein digestion? Why we have to wait for 1.5 days simply by using enzyme denaturation, again alkylation, reduction? Why we need this many steps to confirm one protein sequence? Why can't we confirm intact level? As I mentioned in my previous slides, intact level, when we are saying 150,000 Dalton, I am saying again, it's a 150,000 Dalton. It's a very large molecule. When you are analyzing any large molecules like 66,000, 100 kDa or 150 kDa, there is a chance of mass error, something around 5 Dalton, 4 Dalton, 3 Dalton. Fine. For example, if you take this wall is built by different bricks. These bricks, if you confirm it as a amino acids, these bricks is one line you can consider as a peptides. Okay, this is complete one wall, nothing but your intact protein. If I ask you exactly, can you figure it out? Where is in which position, in which site black color bricks are there, or in which position you can see any breakage of brick, or in which position any repair is happening for this brick, or can you count exactly how many bricks are here? No, that is difficult. Simply, I can say it's a 150,000 Dalton, fine. But if I ask exactly how many kind of amino acids are there, what is the modifications happen here? Can you explain it? No, I can't. Why? Because figuring any small modifications like 2 Dalton difference, 3 Dalton different modification in 150,000 Dalton protein, little challenging. There is no any confidence level when you are confirming any modification which is happening from clone to your resulting protein, any modification happen by 2 Dalton difference, 3 Dalton difference. It is very difficult to confirm intact level. For example, if I remove all these bricks, if I give it as a different bricks, and if I ask, can you check any brick is got damaged, or can you check what is the different kind of bricks you are having? It will be very easy for you. Slowly you can build each one brick, you will arrange it. One peptide you will arrange it. After arranging each peptide you will connect it. You will build complete wall. Here I am having more confidence. Why? Because brick level I checked every amino acid. So I am very much confident when I'm confirming any protein sequence coverage. If I give it as a intact level, in this 150,000, maybe different amino acids modifications may happen. I don't know exactly. When we are discussing in fragmentation level, as I mentioned, A, B, C, D, E, F, G, H, both are looking 100 Daltons, but inside amino acids are different. Same thing will applicable here also. 150,000, yes, it's very clear. I got intact level 150,000. Yes, it's very clear. But when I am asking, can you figure it out? What is the protein? Any modifications are happen? Any PTMs, post-translation modifications happen? It will be little challenging for me intact level. Further, I will do digestion for this protein. I will convert it into peptides. These peptides I will run through mass spec. Then each peptide I will do the fragmentation, means amino acid level. So now step by step, each brick I will build into as a peptide. Each peptide I will connect it, and I will connect it as a complete intact mask, complete wall. So if you are confirming your protein sequence intact level, that is top-down sequencing. If you are breaking as a subunits like light chain, heavy chains, FC regions, like that is subunit level sequence confirmation. If you are breaking, if you are digesting your protein completely into peptides, that is bottom up. So you can clearly see here intact map, middle subunit level, and peptide level, top down sequencing, bottom up sequencing. Why I'm saying multiple times you have to digest the protein, you have to confirm the protein sequence. You can see here, these are the different regions where there is a chance of modifications may happen. You have to confirm your N-terminal sequencing. The same time you have to confirm your any FC regions or FD regions. The same time hinge region, disulfide bonding you have to confirm. So these disulfide bonds you have to confirm. At the same time, any modifications happen on the FC region or FD region, you have to confirm. In this all, by intact level, confirmation is different. But if you do protein digestion and peptide level, if you confirm, that confidence level will be completely different. This is the general protocol when you are doing your protein digestion. Intact map, you will take it. First, you will do the desalting. You will remove complete whatever salts are present inside that, and you will denature the protein. Means normally your protein will have curly structure, like completely mixed up. You will make it as a straight line, as if you will, if you want to analyze with the glycons, you can keep the glycons, or if you want to remove, you can do the deglycosylation, and you can do the reduction completely. Whatever cysteine bonds are there, you will break the bonds. Once you break the bonds, if you leave the protein sample like that, again it may form. For that one, we will do the alkylation. Whatever cysteine bonds are broken, on that we will do alkylation. One alkyl group will go and sit on the cysteine bond, so it won't again recoil, it won't form again this hydrogen bonds. So this is reduction alkylation step, where your 147,000 protein sample is converted into 50,000 or 23,000. Next, protein digestion, where you will add your enzyme trypsin, chymotrypsin. As per your requirement, you will add different enzymes. You will keep it for digestion. Once you keep it for digestion, this whatever protein sequence is there, it will break into, it will break into peptides. By using this peptide, you can confirm N-terminal, C-terminal, amino acid sequence, any PTMs are happen, disulfide bonding you can check, glycan analysis you can do it. This is the one raw spectra I want to show, intact level mass. You can see this is the raw spectra. Your map, any intact level map, if you are analyzing in mass spec, this is the spectra you will get it. Once if you do, this is raw spectra. Why I am saying again and again raw spectra, this raw spectra again you will deconvolute it. Here I am showing as a 2800, 2000, 3000 like that, but I am giving report as a 150,000. This is where I told mass by charge ratio. If it is a 1 plus charge, directly whatever mass you are observing, I can't say as a mass, directly whatever MYZ charge you are observing, that is your mass. For example, if it is a 100 MYZ, with 1 plus charge state, you can consider it as 100 Dalton. What is other charge state? When you are analyzing your samples in mass spec, every time it won't generate only 1 plus charge state. Sometimes it will generate 2 plus, 3 plus, 4 plus, 5 plus. It may go until 50 plus, 60 plus also. By connecting this charge state, as I mentioned in previously also, in mass spec, you are not going to measure your mass directly as a Dalton. You are going to activate your molecule. You are going to give some charge state for your molecule. By calculating the charge state, you will confirm your mass. For example, here I am saying 2800. By charge state, it may be around 24 plus or 34 plus. If your molecule mass is 100 Dalton, if I say it's a 4 plus charge state, in this raw spectra, you are not going to as a 100 MYZ, you will get 25 MYZ. 25 MYZ with 4 plus charge state. That means 25 into 4. My mass is 100 Dalton. I got it as a 4 plus charge state in the mass range of 25. Like this, like when you are analyzing any large molecules directly in your raw spectra, you won't get 150,000 or 130,000 like masses. You will give mass range from 2000 to 5000 or 6000 like that. In this mass range, with this charge state 34 plus, 44 plus like that, your charge state will happen. So this is your raw spectra. When you deconvolute this raw spectra, there you will get your masses directly as your Dalton. Here you can see as a 150,000 Dalton. Same spectra in as a raw spectra. When you are acquiring, you will see it as a range between 2000 to 3500. Okay, this is your deconvoluted spectra. This is the spectra you are going to present in your presentations. Same thing if you do for subunit level. As I mentioned, your instrument should able to match with your theoretical mass. Here I am saying as a 0.52 ppm error. This red color spectra is your experimental data. This blue color spectra is your theoretical data. I am comparing, just I am overlaying, I am confirming. In the presence of 80,000 resolution, you can get 0.52 ppm. If your mass spec resolution is less, automatically your mass error will become more. Same thing you can observe here also. 29 KDA, I am confirming between theoretical, your red color spectra, what are you observing here? That's a theoretical. Here, another one is your, red color is your experimental and this one is your theoretical data. So just I'm overlaying my theoretical data to experimental data and I'm confirming what is the mass accuracy here. This is protein sequence confirmation. Intact level we confirmed, subunit level we confirmed. Now you can observe here closely from E to until R. This is, for example, it's a mapped sample. It's a heavy chain. It's a heavy chain mass. So these small red bricks are nothing but your fragments. From 10 to until 450, this many amino acids, I already done the digestion. I made this heavy chain into peptides. Those peptides went under fragmentation. By doing this fragmentation, I confirmed each and every peptide fragmentation level, and the sequence coverage you can observe here. Sequence coverage is around 99.1. As I mentioned in previous slides also, you can check it from N terminal to C terminal as a B ions, or you can check it from C terminal to N terminal as a Y ions. Overall, you have to confirm every amino acid. That is our main aim here. So here I may miss in Y region or I may miss in B region, but still I confirmed complete heavy chain protein sequence here 99.1%. Why? Because I didn't miss any one amino acid also. I confirmed every peptide here. These are the different spectra of that particular protein sequence here. You can observe clearly B ions, Y ions, it's labeled here. Here our protein characterization is done. Now I will show other applications here. This is a proteomics workflow where you will extract different proteins at a time. Currently, whatever we discussed in our previous slides, that is a single protein characterization. When you want to analyze as a proteome, complete bunch of proteins at a time, you will digest complete proteins in a single epitope tube as a sample. You will generate the peptides. In that case, your instrument should have high scan speed. At a time, you will release number of peptides or more. Still, your instrument should be able to identify every peptide. This is the protein. You are generating the peptides. In LC, it will get separate. Through mass spec, you will confirm the sequence. There are the different steps how you are confirming. This is a LC level. It's a chromatogram where it will be labeled as different site positions. From there, deconvolution. Below you can see as every peptide Y ions, B ions, and you can compare it. It's a complete one chain structure. Beyond this, pharma or biopharma, mass spec is in everywhere. Wherever science is there, their mass spec is there. You can go for proteomics or metabolomics or environment studies, where your river contamination, forensic labs, you can make it. Just 2-3 slides. I kept my product portfolio where our Bruker products you can cover from triple quad, GCMS, LCMS, FTMS, QTOF technology. In worldwide, Bruker QTOF is the high-end mass resolution. We are having 80,000 resolution. We are the person we are having highest resoluted QTOF technology. At the same time, Molotov technology also we are the pioneers in Molotov technology, where it will cover until 10 kHz. Tissue imaging, everything we are doing with our Molotov technologies now. And our recent advanced technology is timsTOF, where ion mobility is, it's a hybrid technology where ion mobility is configured with our QTOF technologies. As I mentioned, MALDI imaging. Recently, we have multiple applications, app notes, publications on Molotov technology. Even now, all pharma companies and biopharma companies also for MALDI imaging, they are shifting from confocal microscopies and all to MALDI imaging. These are high-end reflex. We are the people we are having more than 40,000 resolution MALDI top technologies in complete worldwide. And this is the difference between 2 kHz and 10 kHz. This 2 kHz and this 10 kHz, where you will acquire complete data in nanoseconds, not even in milliseconds. What is the use of this MALDI imaging analysis? You can check the drug distribution, you can design the drugs, and you can trial instead of killing mice and all, instead of doing multiple operations and all. Through our softwares, by using tissue imaging, we can do analysis. You can create your own drugs, distributions, everything. And as I mentioned previously also, Poly2, we are having different tools where you will confirm your polymer samples, which is a little challenging when you are analyzing in LCMS technologies. As I mentioned in the starting of my presentation, Molotov is like a central facility toy. Wherever you want to keep any mass spec facility, Molotov will cover maximum applications of mass spec. Polymers to proteomics, glycomics, biopharma, tissue imaging, microbiology. Even you can identify your bacteria less than 1 minute. Same thing if you want to analyze same bacteria, normally in a traditional way, it will take at least 2 days, 3 days to do the biochemical test, to do the colony, everything. Just you have to keep your colony on the target plate. Directly laser will be applied. You will get direct information as what is the species. Another one is toxicology. Here, even other vendors also, everybody will have technologies are different, solutions are different. When they are giving you as a complete, as a solution ready-made, from columns to complete softwares, instrument, everything, that is called as solution. For example, if you take here toxicology, we are having toxityper as a solution here. From complete sample preparation to until analysis, complete setup will be supplied by Bruker, where LLE, SPE, any precipitation things, extractions, everything will be consumables also supplied from Bruker. So even tools, libraries, everything will be designed the same instrument as a ready-made methods. In this tox type of technology, within 11 minutes, you can run the sample, you can identify the what is the compound. At the same time, you can generate the report. Like this, we are having ready-made technologies. Nothing but tox type of solution, target screener solution. This one is mainly designed for metabolomics and all, where you will acquire the data complete. It will internally connect with online libraries and it will give directly your result as a PDF format. Even for biopharma also, we are having BPC technology, biopharma compass, where data acquisition to until report format as a PDF format, complete process will be taken care by instrument. So now this session is open for QA. Hope I covered in less time. Yeah. Oh, sorry, I taken more time, 10 minutes more. This session is open for QA.
If any participant has questions, please ask.
Regarding instrumentation part or regarding biopharma applications or any, peptide analysis anywhere. In previous also, when you are analyzing any samples and all, if you have any queries, please. I think I got some. Thank you.
Okay, sir. Probably there are no queries. Pandey, sir.
Yeah,
Proceed.
I'll proceed. Okay, so dear participants, we have reached to the end of day here, and this is the last session for today. It gives me actually immense pleasure to thank Shri Venkatesh on behalf of Principal Dr. K.B. Burade sir, our coordinator Dr. Jarde sir, and all other participants. I think Venkatesh sir has explained very nicely regarding the mass instrumentation and its